Okay, so today we're going to be talking about the evolution of eukaryotes and the origin of quote-unquote protists. All right, so we've talked about this already. We've got 5 supergroups. This is specific to domain eukarya. Supergroups do not apply to bacteria or archaea. Just as a reminder, the level of supergroup is going to fall between the level of domain and kingdom. So this is going to be above the level of kingdom. 5 supergroups, which you've met in lab already. We've got SAR, that's a really big one. It's an acronym for Stromatophiles, Alvelates, and Rhizarians. We've got our archaeplastids, that's our red algae, our glaucophytes, our green algae, our charophytes, and our land plants. Excavata, a lot of those are parasites. We saw Giardia, for example, in the lab, but some are free living as well, like Euglena. Amoebozoa, which is going to be our amoeba and our slime molds, at least some of our amoebae. And Epistheconta, which you're familiar with. This is going to include our animals and our fungi and a few other things. All right, this is an even better tree that I like a lot. This is an unrooted tree just for domain eukarya. The arrows in the middle indicate possible places where the root of the tree is. So that's where the root would pull down to where the common ancestor of eukarya and archaea existed. And I like this tree quite a lot because you can see that, well, there's just a lot of groups. There are quite a lot of eukaryotes, most of which you've never heard of. So I'll be referring to this tree as we go through. Don't panic. Of course, you're not going to have to memorize or know anything about all these groups, but I do like to kind of give you the bigger picture of eukaryote divergence. All right, so what is a protist? We know this already, but just to reiterate, it's an informal name for any eukaryote that's not an animal, a plant, or a fungus. And if you've heard of kingdom protista, that is absolutely not accurate. So once again, anytime I hear kingdom protista, it makes me very upset. So if you learned that in 1610, please unlearn that now. We only use protist as an informal name. And protists are extremely diverse. It doesn't really make sense, even morphologically, to throw them together in one kingdom. A lot of them are single-celled, but some are colonial, some are multicellular. And I'm thinking here specifically of like our seaweeds, which are technically protists, not plants. We've got diverse life cycles. We'll talk a little bit about that, but it's pretty varied. And we do have different nutritional strategies. We definitely have photoautotrophs. You can think about diatoms, dinoflagellates, the seaweeds again. Chemoheterotrophs, okay? So this would be similar to an animal where we get our energy and our carbon from organic molecules. And we even have mixotrophs. I love this name, mixotrophs, just like it sounds like to mix it up. So I'm thinking about specifically something like Euglena, which has chloroplasts and does photosynthesize, but also is able to engulf food and eat it. All right, so quick question. How did we go from a prokaryote, shown on the left, that's E. coli, to a eukaryote? All right, couple things to notice about this figure, which I really like. First of all, our eukaryotic cell, and this is an animal cell and not a particularly large one, is quite a bit bigger than our prokaryote, okay? Number 2, we have a nucleus, right? So we have a purple stain in there for DNA. You can see in E. coli, it's kind of divided. That's just a function of where this is cross-sectioned. But just recall, we've got a circular chromosome and it's just exposed to the cell, right? Unlike in the eukaryote, where we have linear chromosomes that are packaged within the nuclear envelope. And then we have a lot of other stuff in there too, and you learned about these organelles in 1610. So question, how did we go from that to that? All right, well, we know this probably happened, I don't know, about 2.1 billion years ago-ish, something like that. These are some early eukaryotic fossils. They are single-celled, as you can see, and lots of extensions coming out of the cell, so you can see they're interacting with their environment quite a lot. But our fossil record, of course, is fairly sparse because these are small, soft things, right? Cells. Individual cells don't fossilize super well, so we do have at least some fossil record, but not a lot. All right, but kind of the real story of eukaryotic evolution is the story of endosymbiosis. And you probably heard about this in 1610. We're going to talk first about primary endosymbiosis, which is what you would have discussed in 1610. And that gives us the mitochondrion and plastids. Okay, chloroplast is a type of plastid, but there are others. All right, mitochondria. They evolved by endosymbiosis of an aerobic prokaryote, specifically an alpha proteobacterium, whereas plastids evolved by a cell engulfing a cyanobacterium. So let's take a look at what that looked like. Okay, first of all, we've got to back up a little bit and start with a prokaryote, because of course eukaryotes evolved from prokaryotes. This is most likely an archaeal cell, and you can see the way I've drawn it. It contains all four of the things that all cells share. We have our plasma membrane on the outside. We've got our cytoplasm inside. We have a genome of DNA, in this case a circular chromosome that's kind of folded up there, and the little black dots, of course, are ribosomes, which we need to synthesize proteins. All right, probably the first thing that happened is we start to get infoldings of the plasma membrane. All right, and they end up giving rise to some of the parts that we know about eukaryotic cells. So at some point we enclose the genome into a nuclear envelope, and at some point those chromosomes become linearized. We also get our whole endomembrane system. So our endoplasmic reticulum, our Golgi apparatus, all the vesicles and vacuoles. And then separate from plasma membrane enfoldings, we're also going to acquire a cytoskeleton. Cytoskeleton would just be long fibers that are made out of proteins. Maybe you recall this from 1610. We have, in order from smallest to largest, our microfilaments or actin filaments, and then our intermediate filaments, and then our microtubules. Okay, all of them just long protein chains. All right, and at this point, we essentially have a proto-eukaryote. It's able to be a little bit bigger because of the infoldings of the plasma membrane. We can sort of think of this as we've increased the surface area over which we can do things, and we've put some of the surface area inside the cell, which allows it to grow larger. All right, but now we're actually going to turn to thinking about our organelles of energy transformation, which would be the plastid and the mitochondrion. All right, this theory of endosymbiosis was popularized by a pretty famous scientist, Dr. Lynn Margulis. The other people kind of had the idea before, but she's really the one that put together the evidence, that advocated for it in the literature. So we think of her as kind of the mother of the theory of endosymbiosis. Fun fact, you might have heard of Carl Sagan, the astrophysicist. They were actually married for a very brief period of time, like, 5 minutes or something. But, of course, they had their own independent careers and trajectories. And she, like I said, this is a quote from her paper, popularized this idea. She said, "The eukaryotic cell is the result of the evolution of ancient symbioses. So symbiosis is living together, and this is the most extreme form." All right, so what does this look like? We have a proto-eukaryote, and here's our alpha proteobacterium. Okay, you can see it's a bacteria. It's got those, 4 things that cells share. It's got its circular chromosome. It looks very similar to alpha proteobacteria today, such as the rickettsias. All right, so primary endosymbiosis is where we have a little Pac-Man looking event here where this is brought inside the cell. And it kind of looks like the larger cell is eating the smaller cell. Maybe that's what it was trying to do. There's a couple of other possibilities, however. It's quite possible that the smaller cell was actually a parasite of the larger cell. And indeed, some of the alpha proteobacteria that we have today, like the rickettsias, are parasites that live within a cell. It's also possible that these cells were already involved in a mutualism, and they just took it one step farther and one decided to live inside the other. All right, so once we have this alpha proteobacterium within the cell, that's not enough to make it an organelle. So then we undergo some changes that make it an integral part of the cell and the mitochondrion that we know and love today. All right, so one thing that happens is we have gene transfer. So part of the bacterial genome actually is going to be moved to the nuclear genome. Most of it is actually, and we're left with a circular chromosome in the mitochondria, about 16,500 base pairs. The rest of it has been moved to the nuclear genome. All right, and some of the eukaryotes that we know and love are cool just with this. So they don't have any plastids, they just stopped with mitochondria. That would include our amoebozoans, our fungi, our animals, some of our excavates. And this is a tree that is showing you the eukaryotic tree of life. At least it's a simplified version. This is one that I made. You've probably seen it in the lab handouts. So here's our opisthecons, animals, and fungi. Those are, technically two different, quote, kingdoms. And then we have a couple of other organisms as well. Choanoflagellates are our closest relatives. Nuclearids are the closest relatives of fungi. Okay, we've got our excavates. So this is supergroup Excavata. You've seen Euglena before in lab. You probably saw some kinetoplastids in lab as well, parabasalids, oxymonads. You might have seen those when we squeezed the termites, and diplomonads. All right, so let's start with supergroup Excavata. What is it? Well, these, like I said, are usually unicellular, generally parasitic, although some are free-living. They're named for a depression in the cell that's called an excavate. You can see it in this figure, kind of like in the middle left of the cell, and this is a feeding apparatus on the ventral surface of the cell. All right, they are unicellular, many of them are flagellated, and many of them, interestingly, have modified or completely reduced their mitochondria. You should have read about that in the background reading. All right, so here's our major groups. We've got our diplomonads, parabasalids, euglenids, and kinetoplastids. We'll start with the diplomonads, and you saw one of these in lab. This is Giardia. Hopefully the figure on the top maybe looks a little bit familiar. To me, it looks like a little teardrop with two eyes. It's unlikely in lab that you would have seen the flagella because it's pretty small, but you should have seen the two, quote, eyes, which of course aren't eyes at all. Those are two nuclei. So in 1610, when we told you that cells have a nucleus, well, again, it was a little bit of an oversimplification. Many cells do have one nucleus, but a lot of cells have more than one, including some of your liver cells, by the way. And our diplomonads are named for that double nucleus. Okay, they do have flagella, like I mentioned, and many of these, including Giardia, are parasitic. Okay, we have mitochondria that have been modified. This is a transmission electron micrograph of a modified mitochondria. You can still see the double membrane, but basically it's not really doing too much anymore. We call it a mitosome, and it is just the remnant of the mitochondria, not really functional in the same way that ours are. So it doesn't do aerobic respiration. Okay, moving on to our next group, the parabasalids. So parabasalids are flagellated as well, but they also have an interesting undulating membrane that allow them to move through viscous fluids. And kind of classic example of a parabasalid would be Trichomonas vaginalis. This feeds on the vaginal lining, and it is the most common cause of what we call yeast infection in the United States, which is funny because it's not a yeast at all. We think maybe it acquired the ability to feed on the vaginal lining by horizontal gene transfer from bacterial species. This is what it looks like in scanning EM, and of course that's been pseudo-colored, not really blue. You can see the flagella and also that undulating membrane. This is what it looks like on a pathology smear. Okay, so if you were to have a yeast infection and go in, they might take a sample and then look at it under a microscope. You can see the eukaryotic cells stained in pink, and then you can see all those little purple cells, which are the Trichomonas vaginalis. All right, these also have reduced mitochondria. We call them hydrogenosomes, and they're able to respirate, but in an anaerobic fashion, and they release hydrogen gas as a byproduct, which is why we call them hydrogenosomes. Oh, whoops, we're going to see those pictures again. All right, moving on to our euglenozoans. All right, so euglenozoans, we have a really interesting morphological feature here, which is a crystalline rod that goes inside the flagella, and we don't know what it does, which is kind of amazing to me. There's still a lot that's unknown about some of these protist groups. All right, this is going to include our kinetoplastids and our euglenids. All right, we'll start with our euglenids. So these are flagellated. They have 1 or 2 flagella coming from one end of the cell. You've seen euglena in the lab, and specifically you've seen one of the species that's a mixotroph. So we have a chloroplast, but it's also able to swim and engulf food, so it functions as a heterotroph as well. This is a video from 1625 of euglena, just to jog your memory. From a former student, and hopefully you remember seeing these. You might have noticed the little red eye spot on euglena, and you can sort of see in here like the pellicle, which is kind of its structure that covers it and allows it to flex like this. Okay, we also have kinetoplastids. So these have also kind of modified their mitochondria to some extent. They've got lots of copies of the mitochondrial genome, and these are heterotrophs. Some of them are free-living and some are parasitic. What you're looking at here are some trypanosomes, which are a kind of kinetoplastid next to red blood cells. And we actually have fossil records for these. So you remember Jurassic Park, I'm sure, where they extracted dinosaur DNA from insects in amber, and we can't really do that. It's too old. But one thing we have seen in these amber insects is that they were carrying these kinetoplastids. They had trypanosomes in some cases. So it's pretty cool that we have a fossil record even of some parasites. All right, most famous one would be Trypanosoma brucei, which is responsible for African sleeping sickness. And there's others as well, so Chagas disease and leishmaniasis. This is what that looks like. It's pretty gross. That's a skin lesion caused by trypanosomes. Now, they're able to evade the immune system because they have this really clever trick that they do. They actually cover the outside of the cell with millions of copies of a single protein. And as soon as the immune system is able to detect and react against that protein, it pulls all of those proteins back into the cell and then switches to millions of copies of a different protein. And it can keep doing this to evade the human immune system. And these are mobile, so take a look at these trypanosomes. Imagine those crawling around in your bloodstream. Okay, moving on to our amoebozoans. So let's talk a little bit about them. This would be some of our amoebae and our slime molds. All right, amoebozoans, this group is probably monophyletic. Okay, so we think it's a pretty good group. And this would include, of course, our classic amoebae, which you've seen in lab. Hopefully you saw a live amoeba and not a dead one. We had Amoeba proteus as well as Chaos carolinensis. All right, so amoebas are very common in soil as well as in water. We have them in the ocean as well as in fresh water. They reproduce asexually. That's a naked amoeba, so it does not have a shell, and you saw that nice cytoplasmic streaming as it extends a pseudopod. All right, so they have, if they're a naked amoebae, they have these pseudopods that they're able to use to move and not necessarily a fixed body shape, okay? It morphs and changes as we look at it. And they are heterotrophic. If you watch them for long enough, you can often see them actually engulfing and eating things like Paramecium, Chylomonas. They're big fans of eating things like ciliates. And you can see that cytoplasmic streaming again here as it pushes that pseudopod forward to move in a particular direction. All right, not all of them are so cute and friendly though. We do have some amoebae that are parasites. So for example, Entamoeba histolytica, which is shown here in its cyst form, causes amoebic dysentery, which you've probably heard of. Dysentery is terrible, terrible diarrhea, and generally dehydration is going to be one of the major problems with that. You're just losing water faster than you can put it back in. This actually kills 100,000 people per year approximately, and many of them are children. All right, so this is not a great thing. We do have it in the United States, although not as commonly as in other places with poor sanitation. All right, moving on to a happier topic, the slime molds. So these used to be classified with fungi, and they do resemble fungi. So you can sort of see why scientists would have thought that. But when we started sequencing genomes, we realized that actually they're not closely related at all to fungi, and they belong with these amoebae. All right, so there's two different types we're going to talk about, the cellular slime molds and the plasmodial slime molds. And they are pretty interesting. Both of these are going to be able to, eat dead organic matter, bacteria, yeast, etc. And they both have the trick that they can differentiate into a spore-forming body, which we call a fruiting body, and then disperse spores into the environment. All right, so let's talk about, I think, the plasmodial slime molds first, and then the cellular slime molds. This is one example. This is the dog vomit slime mold, really beautiful, and it kind of looks like dog vomit, as the name suggests. This you can almost think of as a single cell. It has many, many nuclei. It's huge, and the nuclei aren't separated by plasma membranes on the inside. So this is kind of like a giant multinucleate cell. It crawls very slowly along the ground, consuming food. You can't see it crawl. You have to look at time lapse to actually really see it move, but it can move along the ground. We do have these in Utah. You'll see them sometimes in people's yards, and we do have some down in the desert as well. Maybe not the dog vomit slime mold, but we do have quite a few plasmodial slime molds here. If we look at it in time lapse, you can sort of see it pulsing, right? So you can see it moving. At this point, it's actively metabolizing, not reproducing yet. This is still in the feeding form. All right, in the life cycle, we had the feeding plasmodium, and at the top here, you saw that in the video. And then when the food kind of runs out, it will actually reorganize itself, form a stalk with a sporangium. It will undergo meiosis, release spores, and then these spores will turn into little amoeboid cells and flagellated cells. You can sort of think about them as like egg and sperm. After fertilization, they form a zygote, which starts to divide. At least the nucleus divides, the cell really doesn't, and that gets larger and larger and turns into the feeding plasmodium. Here's another slime mold. I think this one is really cute. And you're going to see in this time-lapse video the reorganization into that fruiting body, into the sporangium. All right, so we're still feeding at this point. It's just kind of living its life. It's kind of a small slime mold. Very cute. I think this one is European. Now you can see those pulsations have slowed down. That's because it's reorganizing itself. You can see that stalk forming now. It wants to move its sporangia up off the floor of the forest here in order to give the spores the best chance to disperse to a different location where hopefully there's more food. All right, so now that stalk is formed. The sporangia are maturing at the top. You can see it's got multiple sporangia, those little amber colored balls. And then eventually it will release those spores, and now you can see it's turned black because it is dying. All right, one more plasmodial slime mold. This is a similar one. I really like this video. You can see that this slime mold is growing on a log, so a fallen tree, and it's like living its life. Metabolizing now, it's actually trying to reorganize itself into that fruiting body. And that's just your daily reminder that everything exists in an ecosystem. All right, let's turn now to the cellular slime molds. So these actually are a little bit different, although they share the characteristic of having a fruiting body. So this is Dictyostelium discoideum. We saw it in the lab. It was on a Petri plate with a really thin thread-like stalks ending in a sporangium. And this is a really important model organism, actually, for studying the evolution of multicellularity, because we have an aggregation of individual amoebae, which are not genetically the same, but they have to cooperate in order to perform this reproductive strategy. All right, so this is kind of a diagram of what it looks like, but I think what I'll do is just explain it as we look at a video. These are several pictures showing the development of this sporangia. That stalk is so thin it almost doesn't seem like it could be supported to me, but it does work. All right, so we have this aggregation of individual amoebae, which form fruiting bodies. And the reason why this is so interesting in terms of multicellularity is that some cells are going to become part of the stalk that holds that sporangium up, and they're never going to get to reproduce. They're essentially sacrificing themselves so other amoebae can reproduce. So it's kind of weird, and how they determine which cell is the stalk and which is the sporangia is interesting, and it's something that scientists are studying. All right, so these ones die without reproducing, but some cells become the spore cells, which are able to reproduce. But they do have some cell-cell communication, and there's some, selection. All right, so here we're looking at a nice video. We're going to take a look at these cellular slime molds under a microscope, and you can see this video might have been made in the 80s by this, outfit and the hair. All right, so these just live in the soil, and that's what we're seeing here. We're looking at a time lapse. Okay, putting it in a Petri plate, looking at under a very dated microscope. I remember having hair like that. All right, and then here we're seeing the individual amoebae. You can see some root fibers or fungal fibers there as well, and they're just sort of, living their life. But at some point, especially if food is in short supply, they're going to start sending out chemical signals that tell all of the amoebae to aggregate, which is what's happening here. So you can see they're actually joining together. We have cell-cell adhesion happening between these amoebae, which again are not genetically identical. And then this mass turns into a slug-like form. We call it a grex, and it starts to move. It starts to move to find a location that it thinks, quote unquote, is going to be good for stalk and sporangia formation. All right, you can see we have aggregations of cells that are joining this slug-like structure. So we're seeing lots of aggregation here. Here's another view. This is a different slug-like form that is coming together. Again, aggregation of individual genetically distinct cells. All right, now we have these little slug-like structures that are going to take off and move to a different location. Isn't that wild? You can tell not only do we have cell-cell adhesion, but we've got a lot of cell-cell communication and coordination in order for this to move. Totally blows my mind. All right, now we're kind of looking at this slug structure from the side, and it's now going to remodel itself, reorganize, and turn into that fruiting body. All right, a little more aggregation. Another slug. All right, and here we go. Now, once again, we're looking from the side. We're seeing that remodeling. We're seeing it stretching out on that stalk, sporangia forming at the tip. And once again, this is what you saw in lab. You were looking at this structure from the top. Incredibly long and thin stalk, spores forming at the top. Okay, so pretty fascinating, right? I mean, it's a little bit crazy, and these have been studied in the lab for a long time, and it's been noticed that there are some dictyostelium cells that have evolved the ability to essentially become cheaters and always make their way to the spore cells. But there's a bit of a coevolution taking place because non-cheaters can recognize these cheaters and tend not to aggregate with them. So there's definitely some sophisticated, chemical evolution that is happening here between these different groups of amoebae. Okay, so like I mentioned, we also have some groups of eukaryotes that have plastids, and these evolve from primary endosymbiosis of a cyanobacterium. All right, so we've got a eukaryote already. It's got its mitochondria. That definitely came first because all eukaryotes have mitochondria, or at least at some point in their evolutionary history had mitochondria. And then here's our cyanobacterium. So we've got, once again, our circular genome, our ribosomes, we've got our thylakoid membranes that allow it to photosynthesize. All right, looks very similar to single-celled cyanobacterium that we have today. And the process is going to be the same. We've got this little Pac-Man event where our cell is going to be engulfed, and same process of turning it from an endosymbiont into an organelle. So we transfer some of the bacterial genome into the nuclear genome. And eventually this becomes our chloroplast. All right, and this is going to include the groups that result from this primary endosymbiosis event. That's going to include our archaeplastids, which are our glaucophytes, our red algae, our green algae, charophytes, and land plants. All right, so here they are on this tree that I constructed. Our rhodophytes, those are the red algae, the chlorophytes are green algae, streptophytes include our charophytes and our land plants. All right, so just to summarize, here's our two events of primary endosymbiosis. The first one is the mitochondrion. The second one is the chloroplast. All right, what's our evidence for primary endosymbiosis? Well, we have quite a bit. First of all, mitochondria and plastids divide by binary fission. So just like a bacteria, they copy that circular chromosome and then the cell splits in half. They have their own circular genome. We already talked about the mitochondrial genome, but here we're looking at a cell that has been stained for DNA. You can see the nucleus is staining really bright. The chloroplasts are staining as well because they have that circular genome. And this is a map of chloroplast DNA from a particular plant. And you can see it's pretty large, 155,939 base pairs. It still has lots of genes. So it has many genes that are coding for proteins, as well as coding for things like transfer RNAs. The mitochondrion does as well, not as many genes, but it still has it. And the chloroplasts and mitochondria can actually be sequenced, and we can make a phylogenetic tree and see that our mitochondrial DNA falls out really nicely within our alpha proteobacteria, and our chloroplast DNA falls out really nicely within the group of cyanobacteria. Okay, mitochondria and plastids also have the ability to transcribe and translate their own genes. So within the mitochondria or plastid, we have RNA polymerase, and it's able to go ahead and transcribe from the genome, and then they translate their own genes with their very own ribosomes. Just to point out here, we have formal methionine instead of methionine. So we're looking at like the transfer RNAs, and that's something that bacteria have. They have FMET instead of MET, which is eukaryote. Speaking of those ribosomes, the ribosomes of the mitochondria and plastids are super similar to bacterial ribosomes. So they're a little bit smaller, same function, right? We're still doing protein synthesis, a little bit smaller, and have fewer molecules of protein and DNA. All right, let's take a look at the membranes. So you might remember that the chloroplast and mitochondria have double membranes. The inner membrane, that's going to be the original bacterial membrane, and the outer membrane is a remnant of the vacuole that brought it into the cell. Our glaucophyte plastids, by the way, actually contain in their membrane peptidoglycan, which you're going to remember is bacterial. That's a component of the bacterial cell wall. Glaucophytes, as far as I know, are the only ones that retain that peptidoglycan, but it's still there, and that's another piece of evidence for primary endosymbiosis. All right, but now let's look at something kind of wild. In some organisms with chloroplasts, we actually have 3 or 4 membranes that surround the plastid, not the mitochondria, just the chloroplast. So what's up with that? How did that happen? All right, well, here we go to secondary endosymbiosis. So this is where things get a little bit wild. Instead of engulfing a bacterial cell, this is when a eukaryote engulfs another eukaryote, specifically to acquire its plastids. And we also, by the way, have evidence for tertiary or quaternary symbiosis, which is kind of wild as well. But, once we engulf that cell, pretty much everything is extraneous other than the chloroplast. In some organisms, we also retain part of the nucleus, which becomes what we call a nucleomorph. All right, so what does this look like? We've got our eukaryote. Here's our photosynthetic eukaryote. And look, it's got all this stuff, right? It's got a nucleus, an endomembrane system, cytoskeleton, mitochondria, and chloroplasts. And now this larger cell is just going to go ahead and engulf the whole thing. Notice how we already have like a double membrane around the chloroplast. Now we've got 2 membranes around the cell. And we're going to lose most of those structures. So we started with all these cell parts, don't really need them. They're going to get degraded. In some organisms, the nucleomorph is retained, sometimes even with DNA. But in most species, we're actually going to lose that completely. And now we're going to end up with a plastid that has 4 membranes. In some cases, one of those membranes gets degraded and we end up with 3. And this is going to give rise to some of the organisms we've already seen and know and love in lab. Okay, so we're thinking mostly here about SAR, but also we're going to take, a quick trip to Euglenozoa as well. All right, so these are all going to be results of secondary endosymbiosis. Our brown algae, diatoms, golden algae, and this is our big group right here of SAR. Okay, so stromatophiles, alveolates, and rhizarians. So this essentially is a group that we put together by molecular similarities, in other words, DNA sequencing. There's no morphological synapomorphies that we can really point to. All right, so let's start with our stromatophiles. We have more than 100,000 species of these. Originally, these were classified because they have 2 flagella of different lengths at some point in their life cycle, but it's not true of all stromatophiles. But you're looking at it here. We've got a smooth flagella and a hairy flagellum, a little tinsel flagellum, and a lot of these are going to be able to photosynthesize. Okay, so for example, we have the water molds, also called the oomycetes. These are another group that looks a lot like fungi. They have filamentous hyphae, but they're not closely related to fungi. If we look biochemically, they have a cellulose cell wall instead of the chitin cell wall that fungi have. Their reproductive cells are flagellated. That's something that everything except maybe the chytrids and fungi have lost, and they have diploid nuclei. All right, you might have seen these if you've ever seen something decomposing in water, like hairy mold-like stuff that covers it. That is actually going to be the oomycete, the water mold. These don't have plastids. They are heterotrophs, and most of them decompose things. We also have to mention a specific oomycete called Phytophthora infestans. This causes potato blight. Here you can see the organism at the top and a potato that's been infected with it at the bottom. And this was actually responsible for the Irish potato famine that happened in the mid-1800s. This left about 1 million Irish people dead, and another 1 million actually emigrated to places like the United States of America. This is a statue in Dublin sort of commemorating this great potato famine of the 1800s. And if you have Irish ancestry and you live in the United States, there's a good chance that your ancestors emigrated from Ireland around this time to escape the complete lack of food. And by the way, the population of Ireland has actually not recovered to the pre-potato famine levels. There are fewer people in Ireland now than there were before 1845. All right, in this group, we also have diatoms. So these are unicellular algae. They have a wall that's made out of silicon dioxide, which is literally glass. And you've seen lots of diatoms most likely in the lab, especially if you're doing the water biodiversity project. Okay, so these are photosynthetic organisms, and they're pretty abundant in freshwater, as some of you have noticed. And they're also abundant in phytoplankton in the ocean. Some are even symbiotic. Okay, here's a video of them moving. You can see Oscillatoria is here, as well as lots of diatoms. Okay, let's move on to another stromatopile, our golden algae. These are mostly photoautotrophs. Some are mixotrophs. They can be colonial or unicellular. We're looking at two colonial species here. Dinobrion on the left and Cynura on the right. Generally, cells have two flagella. These can be really pretty. I really like this picture on the left of a colonial diatom, but they can also be single-celled, and we can have blooms as well, which is what you're seeing on the right. This is a golden algae bloom off the coast of Japan. These can be important in freshwater habitats, and they make up some of the plankton as well, the phytoplankton in the ocean. All right, brown algae, you're familiar with this if you've ever been to one of our coasts, probably. These are large, complex, multicellular seaweeds. They have chloroplasts with 4 membranes, which they use to photosynthesize. All right, why are they brown? Well, they do have chlorophyll A and B, just like plants, so you might think they would be green, but they have another pigment as well called fucoxanthin, which turns them brown. Their cell walls contain cellulose as well as alginic acid, and these are actually harvested and used as an emulsifier in foods and other products. Okay, so we're looking here at a nice seaweed. These, again, have differentiated tissues. We don't call them the same names as plants because they're not technically vascular. So instead of a leaf, we call it a blade. Instead of a stem, we call it a stipe. And instead of roots, we call it a holdfast. All right, these exhibit alternation of generations, just like plants, and we'll probably get to that a little bit later. All right, moving on now to our alveolates. This is probably a monophyletic group. They are named for small membrane-bound vesicles called alveoli in a layer that are just under the plasma membrane. And they weirdly have linear mitochondrial DNA, which is kind of profoundly weird for anything, right? Because mitochondrial DNA is usually circular. All right, alveolates. We've got dinoflagellates, which we've seen in lab before. These have 2 flagella, one that likes to wrap around the meridian of the cell and one that kind of propels it forward. They have cellulose plates for armor, and these 2 flagella give it kind of this characteristic motion that you've probably seen before. A lot of these are photoautotrophs, some are heterotrophic, and some are mixotrophs. Okay, dinoflagellates, really important. They are a major component of phytoplankton, but also they are mutualist with coral. In the coral, we call them zooxanthellae, and they actually make a good portion of the coral's food. So you might have heard about coral bleaching. This occurs when we have either warming ocean temperatures or acidification. The coral gets stressed and it actually either expels or even eats the dinoflagellates that are its symbionts. Once it does that, it actually loses its coral and becomes white looking because now we're seeing the calcium carbonate skeleton of the coral underneath. Coral bleaching is reversible if the zooxanthellae can come back quickly enough, but it can also become permanent, in which case the coral will die. And this is a major problem right now, as I'm sure you're aware, in our oceans. Dinoflagellates are also responsible for red tides, which you might have heard of. And this is actually kind of cool because red tides often look a little like this because dinoflagellates, at least many of them, are bioluminescent. So this is a guy who is out at night surfing a red tide, and you can see the dinoflagellates that are lighting up as he does so, as they're disturbed by his swimming and surfing. If you've ever been to a bioluminescent bay, like in Puerto Rico, that also is caused by dinoflagellates. Okay, so some alveolates are not so awesome though. We have apicomplexans, which are generally animal parasites. These are named for their apical complex, which is specialized for boring into host tissue. These do have plastids, but they've lost the ability to photosynthesize, so those plastids are no longer active. And the most famous one would be the protist that causes malaria. So we have 4 different species of the genus Plasmodium that are able to cause this. Between 500,000 and 1 million people die each year from malaria. And this is transmitted, as you're probably aware, by mosquitoes. But it's not mosquitoes that are the real issue. It's the protist that they're carrying. We have other apicomplexans as well that infect animals. Pneumocystis carinii is one example. It causes pneumonia, but you might be more familiar with Toxoplasma gondii. So this is a parasite that's actually able to alter its host's nervous system and change its behavior. So it goes through one host, which is generally a rodent or a bird. And rodents and birds that are infected with this tend to, in fact, lose their fear of other animals. And that is important to the protist life cycle because it wants to return to a cat, which is its other host. So this kind of behavior makes it more likely that it would be eaten by a cat. You might have heard of toxoplasmosis. Humans can get this parasite as well. And generally, it's not going to kill them, although there are some suggestions that it can alter human behavior as well. Okay, next we have the ciliates. That's something that you've seen in lab as well. These are named for their cilia. Paramecium would be one example. You can see the cilia beating on the bottom left-hand side. And some use their cilia to move from place to place. Others are using cilia to feed. So for example, these Vorticella, which you might have seen in lab, the cilia are beating in order to direct food into the organism, and then they actually contract and will digest it. So you could see that happening right there. Okay, so let's move on to our rhizarians. So rhizarians, they are some amoebae, actually. They have tests, and our most famous ones are going to be the radiolarians and the foraminiferans, but we'll talk really briefly about the chlorarachniophytes as well. So they're down here at the bottom. Radiolarians, like diatoms, have a skeleton made of silica, but in this case, they actually keep it on the inside. You saw these in lab. They're glassy, really beautiful, with these perforations that allow the pseudopods to extend. Here's a living one with the pseudopods actually deployed. They're able to stick to prey organisms and then bring those into the cell. Okay, our foraminiferans, you saw these in lab as well. So these have tests that are made of calcium carbonate. Some of these look like little shells, right? And the pseudopodia will come out through pores in that test. And we have many of these in the fossil record. They actually make up the White Cliffs of Dover, as well, which is in the UK, as well as most of the world's limestone and marble. All right, and then finally we have the chlorarachniophytes. These are named because they kind of look like spiders. They have these spidery extensions. These are amoebae, but they're also photosynthetic, and they acquired that ability from secondary endosymbiosis of a green algae. And remember when I mentioned the nucleomorph, these actually do have nucleomorphs, so the remnants of a nucleus. And we want to mention, and we're going to end with this by talking about one in particular. This one is called Pollinella chromatophora. It's an autotroph, and it has a plastid that is actually unique because this plastid, once we sequence the DNA, we realize that it evolved from a different cyanobacterium than the plastids of every other photosynthetic eukaryote that we have looked at. So we already talked about 2 examples of primary endosymbiosis, the mitochondrion and the plastids, and this is actually a 3rd example. So it's pretty wild, and it makes me wonder if there's other examples out there of primary endosymbiosis that we're just not aware of yet. Okay, that's it for now. See you next time.